Bismillahirrahmanirrahim. Assalamu alaikum. Today we are going to start with the second lecture of gastrointestinal tract physiology. And what is that about? That is about the neural control of gastrointestinal function. This is very important to know that what is the enteric nervous system. So we are going to learn about this enteric nervous system today. The gastrointestinal tract has a nervous system which is of unique type, and it is all its own type of the nervous system inside the gut, which has been given the name of enteric nervous system. Now, what is it actually? That it entirely lies in the wall of the gut, starting from the esophagus and extending all the way down till the anus. The number of neurons in this enteric system is about 100 million. So we can say that 100 million neurons, they are combining together to give rise to this unique type of enteric nervous system inside the gut. And these neurons, they are almost exactly equal to the number in the entire spinal cord. Now, this highly developed enteric nervous system is especially important in controlling the gastrointestinal movements and secretion. Now, let's talk about the plexus of enteric nervous system. The enteric nervous system is composed mainly of two plexuses. Now, one is an outer plexus and the other one is the inner plexus. The outer plexus is lying between the two muscle layers of the gut. And what are they? They are the longitudinal and muscle layers. And due to the lying of this nervous system between the muscle layers, it has been given the name of Myenteric plexus or Orbach's plexus. Now, there is presence of an inner plexus as well, which has been given the name of submucosal plexus or Meissner's plexus, which is lying beneath these, which is lying inside the submucosa. Now, there are nervous connections between them and within them. So we can say that this enteric nervous system, which is divided into two portions, they are in connection with one another. And the myenteric plexus is controlling the gastrointestinal, or you can say the gut movements. And the submucosal plexus is mainly controlling the gastrointestinal secretions as well as the local blood flow. There is a sympathetic and parasympathetic nerve supply as well, which is connecting to both the myenteric and submucosal plexuses. Now, stimulation by the parasympathetic and sympathetic systems, they can greatly enhance or inhibit the gastrointestinal functions. Now, let's talk about the plexus of enteric nervous system. See, what is it? There is connection between the myenteric plexus, among them the neurons themselves, as well as with that of the neurons of the submucosal plexus. And at the same time, they are having the connections with that of the autonomic nervous system, that is both with that of the parasympathetic and sympathetic nerve supply. And the sensory neurons, they are actually going to the prevertebral ganglia, spinal cord, and the brainstem as well. So this is about the plexus of enteric nervous system. You should all know about this. Now let's talk about the plexus in detail. The sensory nerve endings, which originate in the gut, send the efferent fibers to both the plexuses of enteric system, to the prevertebral ganglia of sympathetic nervous system, to the spinal cord, in the vagus nerves, all the way to the brainstem. These sensory nerves can elicit local reflexes within the gut wall itself and still other reflexes that are relayed to the gut from either the prevertebral ganglia or the basal regions of the brain. Now, we should be knowing about the differences which do lie among the myenteric and submucosal plexuses. So, we should be knowing about them. So, myenteric is consisting mostly of a linear chain of many interconnecting neurons that extends the entire length of the gut, whereas the submucosal plexus is mainly concerned with controlling function within the inner wall of each minute segment of the gut. Now, myenteric plexus is concerned mainly with controlling the muscle activity along the gut length, whereas the submucosal plexus is actually responsible for the gut secretions as well as the blood supply of the gut. Whereas myenteric plexus is going to increase the tonic contraction, or you can say the tone of the gut wall, right? So these are the basic differences which do lie among the myenteric and submucosal plexus. Some of them are like, some of the neurons, they are inhibitory, and their fibers, they secrete an inhibitory transmitter, possibly the VIP, that is the vasoactive intestinal polypeptide, or some other inhibitory peptide. Whereas in submucosal plexus, the control the local gut secretion, local absorption, local contraction. So they have the various degrees of infoldings of gut mucosa. Then there is increased intensity of rhythmical contractions is the task to be done by the myenteric plexus, and they're going to have the ability to increase the rate of rhythm of contraction. They have the ability to increase velocity of conduction of excitatory waves along the gut wall, and is causing more rapid movement of gut peristaltic waves. Now, when we are talking about the nervous system of the gut, we should be knowing about different neurotransmitters related to gut as well. So we should be starting from the top, that is the acetylcholine, norepinephrine, adenosine triphosphate, serotonin, cholecystokinin, substance P, vasoactive intestinal polypeptide, somatostatin, leuenkephalin, metenkephalin, bombacin. C. The gastrin, cholecystokinin, secretin, rallin, motilin, GIP, GLB1. Again, autonomic control of the gut. C. What is the role on the gut? That parasympathetic division is going to increase the motility, going to increase the release of different secretions of the gut, whereas the sympathetic nervous system is actually decreasing. One of the system is having thoracolumbar outflow, and the other one, that is the parasympathetic one, is having the craniosacral outflow. Now, autonomic control of the gut. Parasympathetic stimulation increases activity of the enteric nervous system. The cranial parasympathetic nerve fibers, they are almost entirely in the vagus nerves. These fibers, they provide extensive innervation to the esophagus, stomach, and pancreas, and somewhat less to the intestines down through the first half of the large gut. Sacral divisions. The sacral parasympathetics originate in the second, third, and fourth sacral segments of spinal cord and they pass through the pelvic nerves to the distal half of the large gut and all the way down to anus. The sigmoidal, rectal, and anal regions are considerably better supplied with parasympathetic fibers than are the other intestinal areas. These fibers they function especially to execute the defecation reflexes. Stimulation of sacral parasympathetic nerves they cause general increase in activity of the entire enteric nervous system. Sympathetic stimulation usually inhibits the gastrointestinal tract activity. The sympathetic fibers to the GIT originate in the spinal cord between segments T5 and L2. Most of the preganglionic fibers which originate, or you can say which innervate the gut after leaving the cord, they enter the sympathetic chains which lie lateral to the spinal column, and many of these fibers then pass on through the chains to outlying ganglia such as to the celiac ganglion and various Myenteric ganglion. Postganglionic fibers then spread through the postganglionic sympathetic nerves to all parts of the gut. The sympathetics innervate essentially all of the gut rather than being more extensive nearest the oral cavity and anus. And as is true of the parasympathetics, the sympathetic nerve endings they secrete mainly norepinephrine but also small amounts of epinephrine. Stimulation of the sympathetic nervous system exerts its effects in two ways. That is, to slight extent by direct effect of secreted norepinephrine to inhibit intestinal tract smooth muscle, except the mucosal muscle which it excites. To a major extent by an inhibitory effect of norepinephrine on the neurons of the entire enteric nervous system. Strong stimulation of the sympathetic system can inhibit motor movements of the gut so greatly that this can literally block movement of food through the gut. Now, afferent sensory nerve fibers from the gut. Many afferent sensory nerve fibers they innervate the gut. The sensory nerves they can be stimulated by the irritation of the gut mucosa, by presence of specific chemical substances in the gut, by excessive distension of the gut. Then the signals they are transmitted via fibers and can cause the excitation or under other conditions can lead to inhibition of intestinal movements or intestinal secretions. Now, other sensory signals from the gut go all the way to multiple areas of the spinal cord and even the brainstem. For example, 80% of the nerve fibers in the vagus nerves are afferent rather than efferent. These afferent fibers they transmit sensory signals from the gut into the brain medulla, which in turn initiates vagal reflex signals that return to gut to control many of its functions. The anatomical arrangement of the enteric nervous system and its connections with the sympathetic and parasympathetic systems they support three types of gastrointestinal reflexes which are essential to gastrointestinal control. They are the following: reflexes that are integrated entirely within the gut-walled enteric nervous system. These include reflexes which control much gastrointestinal secretion, peristalsis, mixing, contractions, local inhibitory effects, and so forth. Now, reflexes from the gut to prevertebral sympathetic ganglia and then back to GIT. These reflexes transmit signals long distances to the other areas of gastrointestinal tract, like number 1, the gastrocolic reflex. Number 2, enterogastric reflex. Number 3, the colono-ileal reflex. So, the signals from the stomach to cause evacuation of colon, the gastrocolic reflex. Signals from the colon and small gut to inhibit the stomach motility and stomach secretion, the enterogastric reflexes. Reflexes from the colon to inhibit emptying of ileal contents into the colon, the colono-ileal reflex. Reflexes from stomach and duodenum to brainstem and back to stomach by way of the vagus nerves to control the gastric motor and secretory activity. When pain reflexes, they can lead to general inhibition of entire gastrointestinal tract. Defecation reflexes, which travel from colon and rectum to spinal cord and back again to produce the powerful colonic, rectal, and abdominal contractions required for the defecation. The defecation reflexes. Now let's talk about the hormonal control of gut motility. The gastrointestinal hormones, they are released into the portal circulation and they exert physiological actions on target cells with specific receptors for the hormone. The effects of the hormones persist even after all nervous connections between the site of release and the site of action have been severed. Let's talk about them one by one. First, you have to enlist the hormones, which are gastrin, cholecystokinin, secretin, gastric inhibitory peptide, mortallin. So let's talk about them one by one. It is very frequently asked viva question that enumerate the different hormones related to gut, and you are supposed to start with that of gastrin. Gastrin, the stimulus for its release is the protein as well as the distension, and is released by the G cells of the antrum of stomach, duodenum, and jejunum. And its function is to stimulate the gastric acid secretion, and it is inhibited actually by the acid release. Then we have the cholecystokinin. The stimulus for its release is protein, fat, and acid, and the site of secretion is the I cells of the duodenum, jejunum, and ileum, and it stimulates the pancreatic enzyme secretion, pancreatic bicarbonate secretion, gallbladder contraction, growth of exocrine pancreas, and it inhibits the gastric emptying. Now another hormone which is very important, and what is that? That is secretin, is being released as a stimulus of fat by the S cells of duodenum, jejunum, and ileum, and it stimulates the pepsin secretion, pancreatic bicarbonate secretion, biliary bicarbonate secretion, growth of exocrine pancreas, and inhibits the gastric acid secretion. Gastric inhibitory peptide. The stimulus is protein, fat, carbohydrate. Stimulated is going to stimulate the K cells of duodenum and jejunum, stimulates the insulin release, and inhibits the gastric acid secretion. Mortallin is the stimulus is fat, acid, or the nerve, and released by the M cells of the duodenum and jejunum, and it stimulates the gastric motility, intestinal motility. Now, gastrin is secreted by the G cells of the antrum of stomach in response to the stimuli associated with ingestion of a meal, such as distension of stomach, the products of proteins, and gastrin-releasing peptide released by the nerves of gastric mucosa during vagal stimulation. The primary actions of gastrin are stimulation of gastric acid secretion, stimulation of growth of gastric mucosa. Cholecystokinin is secreted by the eye cells in the mucosa of duodenum and jejunum, mainly in response to digestive products of fat, fatty acids, and monoglycerides in the intestinal contents. This hormone strongly contracts the gallbladder, expelling bile into the small intestine, where the bile in turn plays important roles in emulsifying fatty substances and allowing them to be digested and absorbed. Cholecystokinin also inhibits stomach contraction moderately. Therefore, at the same time that this hormone causing emptying of gallbladder, it also slows the emptying of food from stomach to give adequate time for digestion of the fats in the upper intestinal tract. Cholecystokinin also inhibits appetite to prevent overeating during meals by stimulating sensory afferent nerve fibers in the duodenum. And these fibers, in turn, they send signals by way of the vagus nerve to inhibit feeding centers in the brain. Secretin was the first gastrointestinal hormone which was discovered, secreted by the S cells in the mucosa of duodenum in response to gastric acid emptying into the duodenum from the pylorus of the stomach. Secretin has a mild effect on the motility of gastrointestinal tract, and it acts to promote pancreatic secretion of bicarbonate, which in turn helps to neutralize the acid in the small intestine. Gastric inhibitory peptide GIP is secreted by the mucosa of the upper small intestine, mainly in response to fatty acids and amino acids, but to a lesser extent in response to carbohydrate. It has a mild effect in decreasing motor activity of the stomach and therefore slows emptying of gastric contents into the duodenum when the upper small intestine is already overloaded with food products. GIP, gastric inhibitory peptide, at blood levels even lower than those needed to inhibit gastric motility, also stimulates insulin secretion and for this reason is also known as glucose-dependent insulinotropic peptide. Mortallin is secreted by the stomach and upper duodenum during fasting, and also known function for this hormone is to increase the gastrointestinal motility. Mortallin is released cyclically and it stimulates the waves of gut motility known as interdigestive myoelectric complexes, which move through the stomach and small gut every 90 minutes in a fasted person. Mortallin secretion is inhibited after ingestion by mechanisms which are not fully understood. So today's lecture is very important regarding the enteric nervous system. We should be knowing about the nervous control as well as the hormonal control if we want to learn about the gut functions, how the process of secretion, digestion, absorption that is taking place, and what are the different and how the nervous system of the gut is quite different from the rest of the body. So you all are very much desire to learn it in detail, and so that we should be knowing about the functioning of rest of the gut easily. So stay blessed and Allah Hafiz.